generates various reduced oxygen species. And the canonical way that mitochondria do that is in cytochrome oxidase, where electrons are put on effectively 4 at a time through the 4 different redox centers in the cytochrome oxidase, and then handed on to oxygen in a complex set of reactions which reduce oxygen by 4 steps to harmless water. And it turns out that cytochrome oxidase is extremely well constructed, and it really doesn't leak electrons. So all you make is water. It's a harmless reaction, and there's really no radical production associated with cytochrome oxidase. However, there are 2 other ways that you can reduce oxygen. You can put electrons effectively in pairs, often one and then another one very quickly, to make hydrogen peroxide. Or you can put them on singly to make superoxide. And these are the 2 forms of reactive oxygen species that I'm going to talk about, and that are the ones that are implicated in diseases and in aging itself. So the question is, how do mitochondria do this, and can we stop them from doing it? This is my diagram of mitochondria and how they work, and it's kind of complicated compared to the nice, friendly blob diagrams that you often see. And that's deliberate. It's to elevate into my consciousness and yours the idea that there are different energy levels within the electron transport chain. So this is 3 planes of isopotential groups arranged according to their redox potential, from the very reducing through the not quite so reducing to the relatively oxidizing, right down to the oxygen-water couple, which is a very oxidizing couple. And the normal mode of electron flow in mitochondria would be that metabolism gathers electrons from the TCA cycle, from beta oxidation, from other catabolic processes, feeds them in through a bunch of different enzymes into the NAD pool, which is at this reducing potential in mitochondria. And those electrons are then fed through complex 1, and they drop down in energy. And as they drop down in energy to this more oxidizing potential, you can harness some of that energy by proton pumping and generate proton motive force, which you then use to drive oxidative phosphorylation. So this is where you get the payoff, is as the electrons drop between the levels, you can harvest energy and use that to drive ATP synthesis. Some of the electrons that you can gather from catabolic reactions are not so reducing. So you can't feed them in the top. They don't have enough energy to hit this energy level. So you pick them up at the next energy level down at the isopotential group, which includes the ubiquinone QH2Q couple. So the classic example of that would be succinate. Succinate dehydrogenase just doesn't have enough thermodynamic push to put the electrons onto the top level, so they come in at this isopotential group. And they join the electrons coming down this waterfall from the group above. They pass through respiratory complex 3. Again, there's energy harvested to make ATP, and then through respiratory complex 4. Eventually, the electrons do the 4-electron reaction to form water from oxygen. And these red dots are the different sites that have been identified by other labs and by our lab at which electrons can leak out of this chain onto oxygen to make superoxide or hydrogen peroxide. And you can see they're scattered all around. They're predominantly in these more reducing parts of the chain, as you would expect. Only when the electrons are very reducing are they going to leak out and react with oxygen. When they're down here, they're much easier to control because they're less reducing, so you get much less superoxide or hydrogen peroxide production. Some of them are in the dehydrogenases that feed in, particularly in the 2-oxoacid dehydrogenases, pyruvate dehydrogenase, or alpha-ketoglutarate dehydrogenase, various other oxoacid dehydrogenases. Some of them are in the dehydrogenases that feed in to this intermediate level at the isopotential group of the quinones, particularly succinate dehydrogenase and glycerol phosphate dehydrogenase, but also the acyl-CoA dehydrogenase of fatty acid oxidation. And others of them are on the respiratory complexes themselves. There are, at least in my depiction of it, 2 different sites in complex 1, one where the flavin binds and one where the quinone binds. And there's some discussion about whether that's 2 sites or 1 site. I think it's 2 different sites. And there's a very important site in complex 3 where the quinone binds. And because there are all these different sites, we need to give them convenient labels so we can talk about them. So I give them names which are, first of all, the name of the complex, 1 or 3, And then secondly, the name of the site where they interact. So site 1F is the flavin site in complex 1. Site 1Q is the quinone binding site in complex 1. Site OF is the flavin site in the 2-oxyglutarate dehydrogenase complex. Site 2F is the flavin site in succinate dehydrogenase. So I'll talk about site 1F, site 1Q, site 3QO, site 2F. And these are the ones I'm referring to, these sites in the different complexes.
Martin, can I ask a question, please?
Please do.
So I thought the